Hi everybody. So in today's video series, I thought I will discuss a particular topic that known as G protein coupled receptor. So I recently gave a presentation about some of the critical function that been revealed, how a class of G protein coupled receptor functions. And today I thought, why don't I make a video about this particular concept, and let's see what they offer. So this particular paper being published, or particular work being published on Cell Journal, it's on 2022, so by a group of scientists. So they are mostly from University School of Medicine, Stanford, California. So and some other few collaborators from other universities, and I think one of them is from Japan. So this is the particular paper, as you can see. So it's a very interesting paper. So these are the few points when I read the paper that I marked, that are I needed to go back to on Google search to learn about them and learn about this topic. So this is a pretty elaborate paper, and I think they did a pretty good job. So initially when you read a paper from these types of journals such as Cells and stuff, it's very convoluted, like all things are too much interconnected. You don't get everything like on one read. You need to read for multiple times to get the core of these types of papers. So as I have to read at least 20 times this paper to prepare my presentation for the seminar. So that's why I thought if I can explain a little bit about this paper, it would be helpful for the viewers who are trying to learn something about this concept. Okay, now let's jump on the presentation. So even though the paper is pretty elaborate and so much data they presented on the presentation, I make it simple. I don't explain all the data. If anyone particularly feel that they need to learn more, or deep dive into the paper, they can always go back to the paper and check by themselves. But this is just to get a preview of what they did, not like I am completely giving a comprehensive presentation of their work. So let's see. So the paper title as Membrane Phosphoinositides Regulate G protein coupled receptor and beta arrestin complex assembly and dynamics. So as I said, this published on Stanford School of Medicine. So this is Dr. Brian who led this particular study. So he was also awarded Nobel Prize in Chemistry in 2012 for his work on G protein coupled receptor. So this would be the typical overview. I'm not going to here. So now let's see what's the G protein coupled receptor. So G protein coupled receptor are evolutionary related proteins that are cell surface receptor, as we know. And this cell surface receptor act like an inbox for message in the form of light energy, peptides, lipids, neurotransmitter, and proteins. So and this G protein coupled receptor interact with G protein that anchors to the plasma membrane. So when an external signal molecule such as a protein or peptide or anything neurotransmitter that binds to a G protein coupled receptor, it causes a conformational change in the receptor, and the change triggers the interaction between the receptor and the G protein. Now in the G protein there's three subunits: alpha, beta, and gamma. In the alpha subunits there's a GTP attached to it. Now when this G protein got stimulated by the receptor, this GDP is replaced by GTP. So now here you can see the GTP is attached to alpha subunit, and as well as the beta and gamma subunit detached from the alpha subunit. Now this is an active form of G protein, and this is an inactive form of G protein. Now after activation of the G protein, it does is downstream works. But here for this paper, our main focus on the receptor, and more specifically the active receptor. So once the G protein receptor got activated, it need to stop, otherwise it will keep activating the nearby G proteins. So to activate this receptor, we need another protein. This is known as arrestin. So arrestin is a small family of protein that is important for regulating signal transduction in G protein coupled receptor. And beta arrestin is a form of arrestin that ubiquitously expressed and function to block G protein coupling receptor and promote G protein coupled receptor trafficking recycling, known as desensitization. And phosphoinositides, which is the main core focus of this paper, is a minority phospholipids of all eukaryotic cellular membranes, and it's play a vital role in all aspects of cell cycle. So you're not going through this. So let's see the interplay between this, like G protein coupled receptor, arrestin, and phosphoinositides. So once the G protein coupled receptor get activated, it's need to deactivate, and that's where the arrestin comes to play. So based on how arrestin binds to the G protein coupled receptor, the G protein coupled receptor can either be classified as a class A G protein coupled receptor or class B G protein coupled receptor. The class A G protein coupled receptor transiently bind with the arrestin, but class B bind very stably. So after the complex formation of G protein coupled receptor and arrestin, both of the complex got trafficked to the endosomes. And in the endosomes, the class A G protein coupled receptor got recycled, and the G protein coupled receptor goes back to the plasma membrane or cell surface. But for class B, in most cases, it got degraded. So in this particular paper, they said phosphoinositides, which is a membrane lipid, as I said previously, play a role in this complex formation, G protein coupled receptor and arrestin complex formation. And they specifically said the phosphoinositides is required for class A G protein coupled receptor complex formation, but for class B, it's not required. So this is the general graphical abstract from their paper. If so here the phosphoinositides, as usual, whatever I said on the last slide is pretty much everything this explained here. So these are the phosphoinositides that they said play a role in this class A G protein coupled receptor, but don't play any role in class B G protein coupled receptor. Okay, so why this G protein coupled receptor and phosphoinositides is important? So G protein coupled receptor are very important because it play a very crucial role in multiple physiological processes, and it's important for potential therapeutic targets or as for drug discovery. It play a role in biosignaling. It involves in multiple diseases, and more specifically that I want to say for the drug discovery. So you can see here for the FDA approved 34% marketed drugs targets G protein coupled receptor. So that's pretty much 1/3. So, and approved drugs target at least 188 G protein coupled receptor among the thousands of G protein coupled receptors that is been discovered so far. And the global sale volume for this FDA approved drug is around over $180 billion annually. So this is a pretty big number in case of market value. So this is also one of the important reason for exploring how this receptor work, and how the researcher can leverage this receptor to target drug discovery. So here one particular assay they used pretty frequently to measure the G protein coupled receptor and beta arrestin recruitment, and for this they use nanobit protein-protein interaction assay. So this is a very interesting assay where the nanoluciferase is an enzyme that got split into a large bit and a small bit fragment. Here you can see, and this large bit and small bit fragment can be fused with the protein of interest, like here, genetically can be engineered. And when this protein of interest got got interaction, this large bit and small bit can also come close approximity, and when they come close, the reconstitution of the active enzyme occur, and it causes a bioluminescence, and this luminescence can be measured. So this is a very very interesting approach to measure this kind of protein interaction, and it's highly sensitive, and it allows for specific detection in a longer period of time. So let's see the results. So this is the results preview before I dive into the actual results. So I thought if I present this preview, that would help the listeners to understand what the authors trying to convey, what message they're trying to convey. So first they tried, beta-arrestin and phosphoinositides binding is important for desensitization of endogenous G protein coupled receptor. Then they say G protein coupled receptors stratify into two groups based on the reliance on phosphoinositide binding for beta-arrestin recruitment. Then they showed G protein coupled receptor phosphorylation effect dependence on membrane phosphoinositides for beta-arrestin recruitment. Then they said phosphoinositide binding stabilizes fully engaged G protein coupled receptor beta-arrestin complex. And finally, they said membrane phosphoinositides are allosteric modulator and promote beta-arrestin activation. Now, in this particular experiment, they showed how phosphoinositides is required for G protein coupled receptor sensitization. So the left side of the slide is the working principle of this assay. So in this particular assay, they used knocked out HEC290 beta-arrestin knocked out HEC293 cells, and they also used wild type beta-arrestin as well as a mutant type beta-arrestin. So this mutant type beta-arrestin does not recruit phosphoinositides to bind to G protein coupled receptor. So in this particular experiment, they stimulated class A G protein coupled receptor beta-2 adrenoceptor with isoprotranol, which is a stimulant, and measured the receptor desensitization in the presence of wild type beta-arrestin and the mutant type beta-arrestin. And here they measured the CMP response. So CMP response is a measurement of receptor desensitization. So here they measured and they found that there's a significant difference between the receptor desensitization between wild type and the mutant type. So as I mentioned, the mutant type doesn't recruit the phosphoinositides to bind to G protein coupled receptor, and which we will show in the next slide. For class A G protein coupled receptor, it is required, phosphoinositides is required. So here in this particular slide, they showed the beta-arrestin translocation to plasma membrane upon stimulation with G protein coupled receptor agonist. So for this, they fused the large bit protein to the plasma membrane using a CAAX motif, and the small bit they fused with the wild type arrestin, with the wild type and the mutant type arrestin. So when this arrestin containing small bit come close to large bit, it will complete the enzyme nanolook and it cause a luminescence. That luminescence can be measured. Now here one thing to be noticed that for class A G protein coupled receptor, phosphoinositides is required for the binding, but for class B it's not required. And another thing, for the mutant beta-arrestin, phosphoinositides is mutant beta-arrestin doesn't recruit the phosphoinositides. Okay, so you can understand the result better. Here you can see for class A, when they did the experiment, they got luminescence signals for the wild type, but they don't get any luminescence signals for the mutant type, because the mutant type doesn't recruit the phosphoinositides, which as a result it's not get trafficked to the membrane. But for class B, as phosphoinositides is not required, in both cases, the wild type and the mutant type, they could measure the signal. So with this particular experiment, they showed the class A G protein coupled receptor require phosphoinositides for functioning, while for class B, phosphoinositide binding is not necessary. So then they showed G protein coupled receptor phosphorylation pattern affect the dependence on phosphoinositide binding by beta-arrestin. So for this, they used NTSR1 receptor and they did phosphorylation in multiple region. So here these red dots are the site of phosphorylation sites. So they did phosphorylation in ICL3, C-tail proximal tail, C-tail distal cluster, and here the point of the mutation. So they generated around 12 construct, and they did the plasma membrane bystander assay, and from there they found that the cluster that classified as class 2, that is similar as class B G protein couple receptor, they didn't lose the function, while the cluster that classified as class 1 or similar as class A, they lose half of their function due to this phosphorylation. So from this paper, from this experiment, they summarize that the phosphorylation affect the binding of the phosphoinositides and beta-arrestin to the receptor, G protein couple receptor. Then in this particular experiment, they showed how phosphoinositide binding stabilizes fully engaged G protein couple receptor to beta-arrestin complex. So for this, they used full-length wild-type and mutant-type phosphoinositides along with C-tail truncated wild-type and mutant-type beta-arrestin, and they used size exclusion chromatography to measure this complex. So, and from this chromatogram, we can see the C-tail truncated wild-type and mutant generate higher signal compared to the full-length wild-type and mutant-type. So here's one thing to be recalled, that the C-tail of the beta-arrestin is a inhibitory tail of the beta-arrestin. So from this experiment, so here's the quantitative view of this particular graph. So from this experiment, they concluded that PIP dictates, C-tail of the beta-arrestin dictates how PIP binds to the beta-arrestin and G protein couple receptor. Finally, they showed phosphoinositide increases the population of active beta-arrestin. So for this, they immobilize the beta-arrestin to the surface and they injected FAB30. And using the surface plasmon resonance assay, they measured the signal. So from this graph, we can see. So this is the base signal of FAB30 only. So FAB30 has affinity for beta-arrestin, and so this second graph is for FAB30 with phosphoinositides. So phosphoinositide is a membrane lipid. So when PIP452P2, it's a derivative of phosphoinositides. So when they injected this with FAB30, they found that there's an increase in active beta-arrestin, and this is B2RPP. It's an active form of beta-arrestin, so obviously it will give higher signal. So based on this 5 particular experiment, there's few more experiments remain that I didn't include here. If anyone interested, they can go back. So based on this interest, we can summarize that these findings are a molecular basis for understanding the phenotypic classification of G protein couple receptor into class A or class B beta-arrestin. It's independent receptor. And in this model, class A G protein couple receptor require detection of membrane phosphoinositides for recruitment to an activated G protein couple receptor. Finally, they explain some of the limitations of their study, such as this study are limited to in-cell or in vitro experiments. And the in vitro experiments uses G protein couple receptor solubilized in detergent, and the soluble derivatives are membrane phosphoinositides also used. So the derivative they use, these are 8 carbon long instead of the 18 carbon long chain. So, and the in-cell experiment generally employ overexpression. So for some particular assay, they overexpressed the protein into HEC290 cells. Finally, they say to integrate the membrane from phosphoinositides, they use the mutant version of phosphoinositides, mutant version of beta-arrestin instead of the actual beta-arrestin disturbance, and also they used that's what they mentioned here. So this is the summary of this particular paper that is published on Cell. So if anyone can interested to learn more about this, they can go check this article. This is free. So as it says, I will say the name again: membrane phosphoinositides regulate G protein couple receptor, beta-arrestin complex assembly and dynamics. So it's a pretty great paper. If you read this one, you obviously will learn something new, and you will get the understanding how this type of particular work can be conducted, and how how much work they put to publish this type of or get these types of results. Okay, so this is not a trivial paper. It is very elaborate and very comprehensive paper. Okay, so thank you. I hope you enjoy this.